And if you're basing it on color, now that I think about it, it's gray and it's gritty. You're not going to do it either. So you're going to have to go based on taste. And I don't know if I can talk you into how good it probably tastes. I'm sure it tastes really good. Dr. Luther, would you enjoy an oyster milkshake with me? I would consume one if you would.
I might. I do love oysters, especially just fresh when you shuck them yourself on the half shell.
I've had lots of those. And in fact, often when we are in the middle of an oyster experiment, we collect oysters from the oyster growers and often get way too many. So I ended up having to find homes for these fresh oysters every day that don't get used. So I will make sure and let everyone in the department know when I have dozens of extra oysters.
Oyster party.
Wow. You know, if you tempt these undergraduates, they might jump in and take that challenge. $25 is $25.
Yeah. Yeah.
I need to video it though, so I can put it up on the webpage.
That's almost a quarter of a book.
All right, who would like to introduce themselves next?
I'm happy to go if that's all right.
Sure.
Awesome. I'm going to share some pictures because I'm a very visual person. So this is a display of some of the stuff that we do in our lab. We study the function and evolution of natural colors. We typically look at birds' eggs because they're a really cool experimental system that lets us ask questions about the diversity of colors on Earth. So whether we're looking at sort of climate variation, what might predict what kind of colors are present in certain places in the world, or what kind of breeding behaviors might affect colors that you might think are maladaptive, like these great tinamous that live in Central America, that have these massive, bright blue, conspicuous eggs in a place where basically parents and babies get gobbled up all the time. So we ask questions about how colors might be affecting selection pressures acting on them. And as ecologists and ecologists that are studying populations, we're interested in the dynamics between multiple populations, like what you see on the screen here, which is a dynamic between the cuckoo and its host species. This bird right here is grabbing one of the eggs and laying one of her own. And it's a sort of dastardly deed where her offspring are going to eject all of the other offspring in this foster parent's nest. And they have to use the visual information present on the egg to be able to determine whether or not that belongs to her or this cuckoo and avoid it. And so it's really great system for understanding decision making. And we basically use this process where we print and create eggs in the lab. We paint them a specified color of some type that'll tell us something about the neural processes used by the bird. We measure them with spectrophotometers. We find birds' nests out in the field. We deploy those eggs that we've created. One of those is actually in this nest here. And we assess how the birds will respond to those types of cues. And when we do this enough, and of course we publish, so that's very important for your undergraduate and graduate experiences. So we make sure that you sort of have a simple pathway towards publication. And when we do this enough, we have a really good idea about how both decision-making, neural processes, and color vision, as well as the colorful traits, have adapted over time. So we work with lots of birds in the field to better understand how this actually functions. And now we're sort of moving on to color signals in general to understand the spatial and temporal aspect of them. And so you're looking here at an anole, and their dewlaps actually move in time and they change in color. And so we're actually using specialized cameras that we're creating to allow us to see them as they would be seen by a female predator or predator in their actual environment and at the way that they're actually using them. They choose to keep things concealed. They choose to reveal them both certain times and certain places. So those are some of the things that we're working on in our lab and the kinds of questions that excite students that are currently in the lab.
That's really cool. I always find your research so colorful.
Pun
I have a
intended.
non-scientific question for you, Dr. Hanley.
Sure.
Since you have a whole crew that are creating and decorating eggs, do you get busier around this time of the year as we approach a particular holiday?
Is it okay to say Easter? I think it is. Like around Easter is very fun in my lab, regardless of what your observance is. that's the time when really everything sort of, hits the fan, if you will. We have to have hundreds and hundreds of eggs painted. And so we've got, we create our own acrylic paints too. So we make our own paints the way artists would, and we can construct them and we paint them. And you couldn't imagine how many times people walk in and think that we're having a holiday party, rather than getting ready for our field season. And so one of my goals in the next couple of years is to get like a really fun student paper that's timed perfectly with that holiday, because I think that would just really take off, on social media. But it hasn't ever worked exactly right, the timing with publications and the review process and all of that. But I'm like gonna try.
I would say that it'd be hard to find the eggs that you make. Impossible for kids to find the eggs that you make.
Not always. We do crazy colors too. So sometimes it tells us a little bit about the way the brain works if you have like chartreuse or like, neon colors or, totally different things. But sometimes it's hard. And that actually came in handy because in some places people will forage for wild eggs. And there's some cool stories about that.
All right, that's pretty awesome. So who would like to go next?
I can go next. I have some slides to share, so I'll do that.
Sure.
Let me see, share my screen. All right, so that's me on the lower right taking blood from a bird in the island of Borneo. So my lab mainly does evolutionary genomics work. I have a few slides to show kind of the type of research that I do here. But the main overarching questions I'd like to address is what are the processes that shape biological diversity that we see today? So this could be geographic processes, spatial processes, ecological processes such as the fundamental niche of the species, the environmental variation, or even the evolutionary history and earth history of that region. So together they combine to affect what we see in terms of the biological diversity. So the question we ask here span a wide range of spatial temporal skills. I did my PhD in on Southeast Asian birds. So a lot of the question asks how tropical birds speciate and diversify in the old world tropics. So using molecular tools and computational analysis, look at timing of divergence, the patterns of divergence, what are the drivers for causing birds to speciate, how they disperse over oceans and across large areas. Also, I look at things at the genomic level. So this is based on the system of a group of birds that are called manakins that are found in South America and Central America. They're really sort of sexy kind of birds that perform really elaborate courtship displays. And these birds, we found what are the genomic and chemical basis of this sexually selected trait. So you can see these two birds here. They're different colors. We found that, for example, the golden neck color is caused by a carotenoid called lutein, and the green belly is caused by melanin plus lutein. So the birds play around all these chemicals to create sexually sort of appealing traits. And using whole genome sequencing, we could identify the genes that are underlying some of these traits. And also, I work at stuff at the metagenomic level, meaning that these are the combination of all the bacteria and archaea genome in the host system. So the group that I look at for this sort of question are grouse. These are birds that eat plants. And a lot of these plant species that they feed on are toxic, so they have phenolics and all kinds of nasty stuff. And the bacteria in their guts help them to digest and detoxify. So some of the questions we're asking include how are chemical pollutants in the soil affect the gut microbiome of these birds, and how that would affect the functional aspect of these bacterial communities. Would they stop sort of trying to detoxify chemicals for this bird? So these are using shotgun sequencing, lots of sequencing to get the genomes of the bacterial genomes. And then finally, just various projects done by students in the lab. They run a whole gamut of species and questions, going from snakes to mammals to bees and so on and so forth. So thank you.
Well, it looks like you work with a lot of departments. Were some of those students were ESP or?
Yeah, they're both a combination of ESP and SSP. So
Cool. All right. Who would like to go next? Dr. Chiari, Dr. Hunt,
I'll talk.
Dr. Luther?
I don't mind.
I can jump. Oh, okay, go ahead, David. I was unable to find my unmute button for
no,
a second.
Kathleen,
Go ahead,
go
David,
for
and
it.
then I'll go after. Sorry?
Oh, well, I don't have anything formal besides words. I didn't put any special slides together. In part because my projects are just so varied, I can't fit it all into one little thing. But I think the common themes in a lot of my research are at the basis, behavioral and ecological adaptations of animals to their environment. So whether that's species interactions, whether that's a bird singing and how it sings differently in different places, or how they respond to the structure of the forest that they happen to live in. And so this happens throughout the Americas, primarily the Amazon and North America, but some other places too. At least before COVID, it was all these places. And because of the extinction crisis that's going on in the world, so many species are going extinct and others on the precipice of extinction. We've done a lot of work recently, the royal we, my graduate students and collaborators and I, on conservation actions and what we can glean from which species are most vulnerable to extinction and how to try and prevent those extinctions. So we've looked a lot also at what human restoration actions and conservation actions have been implemented and where they've been successful and why, to sort of model that, to see if we can come up with some, I don't know, silver bullets of sorts to try and prevent some of these extinctions from occurring. So on an average day in the lab, well, it's been a lot of lab lately. We do a lot with LiDAR and GIS and mapping and species range maps, or song analysis if we have songs we've recorded. But when we're in the field, it's a lot more behavioral observation of birds or using mist nets to catch birds, identify them, maybe take feathers, look at their plumage, their molt, lots of different other aspects, morphological aspects as well.
That's awesome. And you teach a undergraduate class in this topic, right? Ornithology?
I do teach ornithology and animal behavior are the two kind of staples. Yeah.
Dr. Luther, can I ask you a brief question?
Yeah.
What does an urban bird sound different from its rural cousin? I mean, is there a difference there?
Yeah. Well, it depends which species, right? Because we make so much noise in the urban environment compared to rural in general, right? There are rural places that are very loud also, like right next to 95 or 66 or something. But when you get further from the road, it's a bit quieter. It turns out the ones in quieter places tend to have lower frequency songs compared to the ones in the louder places, which tend to be more urban, but it's not exclusive. I mean, like airports that are rural cause a lot of havoc too. That's the one kind of standard thing that most birds shift, but there's some different things depending on the species. Some of them actually choose different notes to sing also. So they can have whole different dialects that come about because they've shifted so much that they don't want to basically compete with all the high energy, intense, low frequency traffic noise.
Wow, thank you.
Yeah.
Can I also ask this question? I know I don't want to delay, but does that select for specific urban dialects like over time?
Yeah, it's a great question. I don't think we know yet, right? So in the city of San Francisco, where I've done a lot of this, there in the 1970s, which this is before my time, but I collaborated with an older gentleman. There were 3 different dialects in the city in the 70s and prior. But as of the 90s, there was only one left. And the one that had the highest minimum frequency and some other trill note stuff was different, took over and basically replaced the other dialects. The birds were still there, but they now spoke just this one dialect. It's almost as if, there was a wave of, I don't know, DC folks spreading throughout Virginia, which is probably most of the rest of Virginia's big fear. And all the rural dialects in Virginia just get lost because everybody speaks Northern Virginian now, right? So that sort of thing has happened. There seems to be a maintenance of different dialects in different areas for some of these. And this is just the birds that learn their songs, that the dialect changes. But for birds that don't learn their songs, and actually this has been in whales, this has been in crickets, frogs, lots of different species, they all seem to make this adjustment where they have a higher pitch song.
And someone in the comments, so birds have dialects. Is that like language dialects or?
Yes, it is. In fact, I'm teaching this animal communication class this semester, and we were just talking about cultural evolution. Just like in people, birds, not all birds, but the ones that learn their songs, so the parrots, hummingbirds, and the little songbirds, passerines, the Austin passerines, they learn their songs, just like whales, dolphin, I just said that, whales, dolphins, and a couple of monkeys, primates, they all learn and pass these on as social learning from generation to generation.
That's
So
really
just like people,
interesting.
culture.
Yeah. All right, Dr. Hunt, would you like to come up?
Okay, I finally found, my Zoom window always gets buried and then I can't find the unmute button. I live near a train station, so apologies for any train horns. So I will also share my screen, and it's really fun just hearing, of course, as soon as my train comes by. It's fun hearing you guys, hearing my colleagues explain their research, because we do have these areas of overlap that I find really exciting, and I'm hoping to be able to work with Dr. Luther and Dr. Hanley and really almost everybody in my department. So I also study I do some bird work and some behavior and a lot of conservation work, like Dr. Luther. So in fact, we're plotting a project together right now. But in general, my angle is more the physiology. So I also am really concerned about or very interested in conservation of endangered species. But the way I usually start in on that question is by looking at the stress physiology of animals that aren't breeding well or that seem to be in decline, and trying to figure out, are they stressed? If so, what's stressing them? Is the stress so severe that it's starting to impact their reproduction? I have a little PowerPoint here, which I will just go through just to give you some visuals of, give you an idea what the projects that I have actually look like. So I actually started out working birds. I now work more on elephants and whales, but I still am really interested in birds. And one of my big goals when I started here at GMU, which seems like just yesterday, was to continue a project that I've done a lot in Northern Alaska. I did years and years of work in the Northern Alaskan tundra, working on this little bird that's shown up here at the top, which is the Lapland longspur. When I started this project, this was what we considered at the time pristine tundra. And it's now clear that nothing is pristine anymore on this planet, and that the tundra is really changing fast. So this site, since I've been working there, has started to become, we call it the shrubification of the tundra. The tundra is starting to turn into a shrub habitat, and there's some trees moving in. There's a tree now on my old field site, which kind of just freaks me out. So I'm really itching to get back up to Toolik and see how these birds, which are tundra specialists, how they're doing. Are they still there? Are they still breeding as well? Are they starting to retreat? And how's their stress and their reproduction looking? So I'm obviously COVID suspended my plans to get back up there because we couldn't, all fieldwork was canceled, so we couldn't travel during COVID. But I'm hoping to bring some GMU students up to Northern Alaska in May and June of 2022. That's my goal now, and take a look at how the Arctic birds are dealing with climate change. That's a project I really want to start, but that is not yet running. So what I'm actually doing right now, I switched gears a bit, and I'm working on Asian and actually also African elephants at the National Zoo and Zoo Maryland. And this is a small pilot project right now with an ESP professor, Dr. Freeman, who is also studying stress and reproduction in endangered species. So we're teaming up on a project to see if we can measure stress and reproductive hormones in elephant toenails and tail hairs. This may seem like a really obscure question, but the idea is that if we can do this, and I think it's going to work because other people have gotten bits of this to work, we may then be able to look at archived hair specimens from like museums and nail specimens from historic populations of elephants and compare them to current populations of elephants and see if their reproductive and stress hormone levels have changed over time. So the National Zoo and Zoo Maryland are collecting samples for us. They started mid-pandemic and they're just archiving all the samples. They're clipping hairs from their elephants. We keep getting emails from them about, can you take belly hairs? Do you need shoulder hair? Like all these specifics about where on the elephant we want these hairs from. And they're trying to figure out how the toenails grow. This is one of these projects where you get into these like weird little questions. So that's ongoing, and I will need GMU students to help analyze these specimens in the lab, probably starting this summer and definitely in the fall. I think we'll have a full year of samples in the fall. So that is going on. Most of my projects, though, are not birds or elephants. Most of my projects are on large whales. And I have a whole set of these projects rolling forward on all the species pictured here. So gray whales off the coast of Oregon, that's a project on living swimming whales where we go out with the whales. I have samples archived from North Atlantic humpbacks that I'm trying to get back to, respiratory vapor samples, actually. I have baleen from North Atlantic right, Southern right, bowhead, fin, and blue whales, and we have a whole bunch of projects we're tackling on them. So the baleen is what they feed with. It is this thing inside their mouth. It's actually, I now realize, it's really compressed hair is what baleen really is. There's a lot of museums that have baleen specimens that they just accumulated over time. And we have recently realized, or me and my students have recently demonstrated, that baleen incorporates hormones as it grows, and baleen grows slowly over many years. So you can take one piece of baleen and reconstruct that whale's past history of like the past 3 to sometimes up to 15 or 20 years, and see its pregnancy cycles and its testosterone cycles if it's a male, and see how stressed it was, and see these patterns over time. So this has become really exciting. You can't get baleen from living whales, so we're relying on specimens that have been collected from dead animals. But I feel like this is a positive thing to do. Like if a whale washes up dead, you ought to be learning all you can from it. So I work with the stranding networks on the east and west coast of the United States. There are these stranding networks that if a whale is found dead, they do a full necropsy, an animal autopsy. And sometimes I go help. It's very stinky, but really fascinating. And so if there's students who are interested, I would love, we'd be on call to run out to any East Coast strandings. And then there's a lot of baleen in my lab already that I just need help with the lab work on. And it's a really sort of wide open field. We're at the stage now where I can give a student a baleen plate from a species that's never been studied and say, here, figure out what the reproductive cycle is of the fin whale. Nobody has ever done this. And an undergrad can tackle this kind of project and get something publishable. So I've got a lot of baleen in the lab actually right now. I also have a joint appointment with the Smithsonian Mason School of Conservation out in Front Royal, which if any students watching this don't know, Mason students can go for a whole semester or for short summer projects. There's something going on in the sound in the background. Somebody's unmuted. Is that me or is that somebody else? I think that's somebody else. I don't know what that sound is. I'm sorry, maybe that is me. I think it is me. It is me. This is my fault. I had a video queued up to show you guys about how we get blow samples from gray whales, and it started auto-playing another video. That was me. Anyway, so I do have this affiliate appointment at Front Royal, and if any Mason students don't know, there's a semester program you can do out there and work with Smithsonian scientists to study the physiology and ecology and behavior of endangered species. It's a fantastic program. It's a resident program. There's a whole dorm there and a dining hall, and you live out there in Front Royal, and it's a beautiful site. So I have just started working more with Front Royal. COVID has stalled a lot of things, but I'm starting up some antelope projects and starting to collaborate on our main wolf project as well. So that's it. I will stop sharing unless you guys want to see this gray whale video, but I'd have to find it again since it went to another video. I'll show you one more thing. I'm sorry, I took up way more than my time, but I just want to show you a piece of baleen. So this is what it looks like. This is baleen from a blue whale. This was a young blue whale who was hit by a ship, whose skeleton is mounted at the New Bedford Whaling Museum in Massachusetts. And I think you can see there's these bristles at the end. So it's really mammalian hair. This is a normal mammalian hair here, and it's just compressed together into this hard part. So this is what they use to filter feed. But if you drill a little powder, I think you could see I've drilled a little area right there. I used that powder from that area to test that I can measure hormones from blue whale baleen, and I can. And I've never looked at the rest of this plate. If a student wants to take this plate and find out if this young male whale had hit sexual maturity or not, we don't know. We could find out from his testosterone patterns. So I have a lot. I have about 15 or 20 plates like this. Some are much bigger, just awaiting interested students. I've taken way more than my time, so I will shut up now. Thank you very much.
Well, thank you. I didn't realize how diverse everything you're doing is.
It's a lot of diverse stuff, but it's all stress and reproduction. There's a theme there of stress physiology and how stress affects reproduction in animals. And so all of these questions, though it's a ton of different types of animals, it's all really about stress and reproduction in the end. I find it really fun to have a variety of projects, but conceptually, it's really the same question, just tackled in a lot of different species.
Awesome, thank you.
Sure.
All right, Dr. Chiari? Chiari? Sorry.
Yes, that's good. That's good. I'm gonna share my screen. I have a very few slides. Okay, so I'm an evolutionary biologist. I didn't even realize I have a T-shirt about evolution meeting that actually didn't happen last year because of COVID. So my background, when I did my PhD, was conservation genetics. But as an evolutionary biologist, I'm fascinated by anything that is variable. I love variation. And so after finishing my PhD, I decided that my background in molecular biology was not enough to understand variation. And so I basically travel around the world and worked in a different context with many different people to basically gain knowledge in analysis of morphological evolution, do genomic stuff, behavior, field work. So I have done a lot of, I'm not young, so this is why I can have done a lot of things. I've done a lot of different things. And so right now in my lab, we mostly work on reptiles and fish. And we have very different projects that look at how animals adapt to human created situation. For example, for fish, we look at how animals that are species that are endangered and raised in captivity get affected once and they are released in the wild, and what is the impact of this adaptation to basically captive condition for when they are released in the wild. We work on using reptiles for cancer research and longevity. We work a lot on color pattern in animals. And some of the things, I don't know if you can see this video in the middle. So this is basically, I work a lot with mathematicians and we model how color pattern, for example, work and evolve depending on different conditions that are genetic condition, developmental condition, but also like ecological condition. And in these slides, you see some of the animals that we use in the lab. We actually have about 50 live geckos in the lab. COVID hit the gecko a bit hard, but they are all fine. We also have project on conservation of sea turtle. We work with partner USGS and in Mexico. So the project have go from theoretical biology to very applied conservation. We also have a lot of projects on turtles, including the Galapagos tortoises and some freshwater species. And so students that join my group can, a lot of time they just started by taking care of the geckos and make sure they are well fed and they are healthy, and then they get like excited about what we do and they start working on different projects that go from behavior to genetics to other things. And right now I have a very I have a fantastic undergraduate who is working on a behavioral project on temperature preference in gecko. And this experimental setup is so complicated. It's 2 months that we are just brainstorming about the experimental setup for this work. And so here, if you want to see a bit of what we do in my lab or see some video of the gecko or what we do in the field, here you have some link. And so this is basically everything. Thank you.
Awesome, thank you. So you kind of have a little overlap with the colors, right? You and Daniel?
NHC, and yeah.
There's so much overlap with everybody. It's really, really cool. Let me think.
In fact, Daniel is basically collaborating with me for one of the project of one of my grad student, and yeah. I mean, there is, and I discussed it with David. I have another project on coloration that involves birds, and I discussed it with David. Sorry, Dr. Luther. I should call everybody doctor. I'm sorry.
Dave's fine.
Everybody's so formal, and I just not call everybody by first name. Sorry. I'm gonna mute myself.
I'm guilty too. Don't worry. So let's go around. Let's talk a little bit. What's a day in the life typically like for you guys? I know Dr. Luther kind of touched on it when he was talking about his research, but what is, like, when you get out of bed in the morning and come to work, what are you guys doing? And this could be an interesting day. This could be a boring day, but we want to know.
I'll jump in. I've got a really sort of seasonal pattern. So there's times when I'm just focused on teaching, but a lot of the species I study have a certain month when things are happening. And so for years now, I've just seen the year in sort of these months of field work. Like May and June, I go to Alaska and I work on birds. August is whale month because the right whales are off the coast of eastern USA at that time, and my southern right whales down in Argentina are calving at that time. So I usually go to either of those places. Fall is sea turtle stranding season, which I didn't even mention in my presentation. So I have sometimes when I'm just like focused on teaching or just lab work, there's lots of lab work and writing of the papers. But then there's these stints of exciting field work where I'll just for 2 weeks I'm just out with the birds or out with the whales, going out every day. So it's really changeable across the course of a year.
I concur. Mine has a very seasonal rhythmic pattern too, at least, when we're not in COVID lockdown stuff. Spring and summer is all about being outside all the time and observing, whether it's locally or in some other continent, the birds and other organisms. We do a lot of camera trapping with mammals too. And then the winter and fall, winter, early spring are really about in the lab, GIS, organizing our data, analyzing it, writing, rewriting. Oh, and meetings.
Well, everybody has meetings, but not as interesting a topic as yours, obviously.
They're all interesting.
Anybody else want to talk about day in their life?
Yeah, I can chime in. I'm kind of like David and Kathleen, or Dr. Luther and Dr. Hunt. That sometimes we go to the field, but in the lab, it's a diverse range of things. A lot of it used to be lab work because the students do all kinds of different species with different genomes. We troubleshoot, getting DNA from different materials. A lot of the things these days, just computation analysis, there's massive amount of data coming out, learning how to analyze the data, writing codes, things like that. I think that's been a lot of the stuff that I wasn't expecting maybe 5 years ago, that how much of computer work that we have to do these days. But still exciting stuff if we go outdoors. But I think the really sort of enriching part is being able to see the results for the first time after you spend, months trying to analyze the data.
Which is different from Dr. Luther and Dr. Hunt, who are primarily in the field and doing the data collection there. You're analyzing the data in the labs. That's bioinformatics, right?
Yeah, that's bioinformatics. I do go out and collect birds in the field. That's the sort of the exciting part, that you go to, native villages in Borneo and different places. But at the end of the day, that's just the beginning of like a years-long process. But I think it used to be that fieldwork took a lot of time, but these days just analyzing data takes just as much, if not longer.
Cool.
So I do a lot of troubleshooting. When you're trying to use living organisms as tools, they don't always want to behave the way a hammer will, in that you strike the hammer and it works every time as long as you strike it right. Using living organisms as tools is finicky. So we do a lot of troubleshooting. I will meet with a student and they will have exhausted what they think is going on, and we'll sit there and have a conversation. And I actually really enjoy these. While yes, I would like things to work, the conversations tend to get theoretical, they get scientific. It's back and forth on what's going on. We get to feed off each other's ideas, and it's kind of a lot of fun. And sometimes even greater answers come out of problems. It can be serendipity sometimes when what you thought was something not working is actually, it is working, just in a way you didn't expect.
That's pretty awesome. I mean, like it's basically taking, all of the boilerplate stuff you learn in the classroom and you're using in the field, and it's almost like you're the Sherlock Holmes of biology. And that's kind of it. When you have that interest and passion, I think that's what can drive you into a career in research or whatever you feel is your. I'm sorry, I lost my thought, but it's still morning. But that kind of led me into my next question. What excites you all about your research? What is driving you right now? What's the coolest thing about your work right now?
Can I go? Yes. Okay, for me, the most absolutely exciting thing is work with people from different fields. I learn, when you are in evolutionary biology, you have this view of things because you just basically work on variation, but then there is a lot of variation out there. And so the fact that I can work with, as I said, mathematician, I just brought a project with a computer scientist in the Volgenau School, and like this for me is so fast. I mean, it's stressful somehow because it brings me out of my comfort zone, and sometimes, often, things at first don't work out and it's a mess. But it's so fascinating because I learn so much, and then the kind of product and understanding that we get of things is so much more multifaceted because you can look at one thing from so many different angles, and then what is somehow obvious for me is absolutely not obvious for everyone else. And like, for example, when you write a paper with somebody who doesn't know anything about your field, things that you use every day because you're so used to like words and so on, they have no idea what it means. And the same for them. And so it's like, it's a continuous learning, and I love it. It's great. Yes.
Thank you. Anybody else? What excites you?
I'll jump in. I am super excited about my whale data right now because we are learning things about whales that nobody has ever known in the entire history of the universe. And there's just something about finding out something new that nobody's ever known before, even if it's a very small thing. So we just were able to use the baleen from the bowhead whales to verify that, or discover that male whales have annual testosterone cycles. And from that, we were able to figure out when and where the bowhead whales breed, which is, and also the length of the gestation, which we've never known. And so we got this little bundle of new information that nobody's known. And there's just something really cool about that moment where you've spent months in the field, and then months in the lab, and then months analyzing, and then months writing, and then there's this moment where you're like, oh, wow, we learned something new. I just find that really exciting.
That is awesome.
I would agree with Dr. Chiari that the confluence of discipline and findings and insights you get from multiple disciplines is really sort of eye-opening. And also the process of like taking years, starting out knowing nothing much about your system, and then discovering more and coming up with more specific questions and hypotheses that you can test. I think that's really rewarding too, because it's just, you kind of see the growth in the knowledge you're kind of digging out from your system.
So let me piggyback off this question because this brought up a good point. I am a new scientist. I'm a new biologist, and I really want to meet people who are interested in my field. How do I get started? How do I network in biology as an undergraduate or even a graduate student?
If I may, especially during the COVID time, I think Twitter is a fantastic place to do it. If you can't physically go to a conference or hang out on campus or things like that, there are plenty of people on Twitter who are doing things that you find interesting. Talk to them, say, wow, that's really neat, or discuss it with them. They will enter into a conversation with you, and then suddenly you are now in that world, and you can post articles that you like and people will start talking to you. So when you can't, and this works even when you can go to conferences, when you can go to biology groups and hang out, or just hang out after class. But I think Twitter is a great way to stay connected and stay interacting with people when you can't physically do it. That being said, I want to highlight Tom Hutchinson, who is here, who often hosted a post-class Taco Tuesday meeting, and I attended a couple of those, and those got very interesting. So that was Tom took it upon himself to create this group that didn't exist by just saying, who here after class wants to go and keep talking?
And it's just those little actions that basically you can just start building up your network. You can start, collaborating on your passions and interests. And does anybody else have any tricks or trades to, start collaborating and start trying to hone in on what you're passionate about?
I have a couple thoughts. I'm sure everybody has many thoughts, actually. And some of these are really obvious, but I find that students sometimes overlook them. One is to actually look through the department websites at GMU and see who is actually working here already. Like, even I'm a faculty member here, and I just discovered there's a faculty member in another department who's doing research I'm really interested in. And I only just learned that, which is kind of ridiculous. And I think the same is probably true of students, that the professors you've taken classes with, but you might not know who else is out there. So just find out who's right here at Mason. For those students interested in wildlife, pay attention to the Smithsonian out in Front Royal. That's a really cool opportunity that is unique to Mason, and it's a world-class research facility with world-class biologists. So a lot of Mason students are kind of unaware those people are out there, eager for students to work with. So explore and see who's right near you and find out what they're doing, and just email them, get in touch. And then another thing I always recommend to students is to look at who is publishing papers in the field you're interested in, and don't just read the paper. Notice who wrote it and email that person. They will be thrilled that somebody read their paper. And often they're looking for students, or they can point you toward other people as well. The first author's email address, or sometimes the last author's, is always right there at the top of the paper, and that's there for that reason, so you can contact people who are doing research you find is interesting. And usually scientists are, like I said, thrilled to find that somebody read their paper and is interested in it.
I would chime in with something maybe obvious too, is to attend seminars. Just go to as many seminars you can attend. as a grad student, Friday afternoon was my seminar time, and it's always exciting because you never know what's going to happen. And Mason is a big campus, so there's ESP, biology, all kinds of different seminars you can go to. I mean, with COVID, it's harder. I would say that if you can watch out for seminars given by job applicants, those are the best, because I've been told that those seminars are the best these people will provide throughout their life. So that's the best talks they will ever give in their life. So look out for those. I think listening to new ideas, how people do their, how people do their research, what trouble they got into, and maybe even asking questions at the end. That's sort of invaluable.
I would also say not to be timid and don't be afraid to ask questions. So sometimes if you're new and you're sort of shy to think that you might be wrong because you're not immersed in the field, you might be afraid to talk to a scientist, but you really shouldn't be afraid to ask questions. And many times somebody that actually doesn't have experience and is not immersed in the dogma of a particular discipline actually can bring in some of the most exciting perspectives. So use that as motivation to remind yourself that just because you new to this does not mean that you can't bring in a really interesting perspective that will generate some cool science. And in fact, it's actually oftentimes the case when you're coming in fresh with a fresh perspective that you could do that in a way that maybe somebody who's immersed in the field can't. So use that as sort of motivation to not be afraid to ask and not be afraid to try. And I find that that's important to remind people about sometimes.
Looks like we have a question, Tom.
Thank you. Sort of a comment and a question. I definitely want to second the, when we have the chance to be in person, go to the seminars. That's how I'm sure Dr. Luther recognizes me from that. I love going to those seminars. I specifically went to Dr. Chiari's seminar when she first presented. Dr. Lim, I specifically have gone to see you present at our seminars, and Dr. Froelich as well. I think that's the first time that I was exposed to you and what you were doing. These are phenomenal opportunities for us as students to find out what our professors are interested in. We don't get a chance to take classes with every single professor, much as we'd like to, but we can definitely be exposed to what moves them. And the question that I had though was in reference to what I had noticed about what all of you guys have been presenting. There's technology to whatever degree. There's technology that you're using to answer different questions, and that's what brought me as an older student to George Mason. There's new technology. It didn't exist when I was, a teenager. I want to learn how to use this technology. What are you all most excited about? What technology, what new things can you do that, sorry, the question. What kind of new tools do you have? What really gets you excited about where you're going next?
I will jump in. I'm always happy to be the first one to jump in. I think I've done that too many times already, but I'll jump in. There's a bunch of new tools I've been using. So I mostly in the lab study hormones. So I do a lot of hormone assays. So I've already done a big change in how I do hormone assays with some new tech that came in actually about 15 years ago now. But there's a whole nother thing happening now in the world of hormones where we can now get hormones out of different sample types than we used to be able to. So instead of just working with blood, I'm now working with baleen and horn and feather and hair and all those sorts of things. So that's one new development. But the thing I'm actually more excited about is that it's now possible to get your hormone information on the animal, but use whole other tools to look at that animal in different ways. And especially drones have turned out to be a really cool new thing to add to field work, to be able to fly over the animals and see them from above. So most of my whale projects now are getting hormone info and doing drone flyovers to assess body condition and behavior of the whale. And that's totally transformed whale field work. And then another whole category is what I'll call the biologgers, which are all sorts of basically little animal Fitbits that you can stick on animals. and figure out their motion, and combine that sometimes with tracking and location where they are in terms of GPS location. And sometimes you can get heart rate and body temperature with these little biologgers. And the biologgers are getting smaller and smaller. So now we can get them on little birds and other species. And that's just, I think of that as sort of the great new thing in my field, is getting drones and the biologgers and the hormones and then classic old school behavior as well, and all in a big package. And GIS info, but I'll stop there and then let somebody else add other sort of landscape scale tools. There's a lot of new tools. And I feel like the really fun thing is building a big project that uses kind of all the new tools all at once with a big bunch of collaborators. That's really exciting. And you learn so much more. You get such a more complete picture that way.
We've got students in our lab. So one of our projects is a National Geographic project that's looking at photography and videography. And so if you can imagine for a moment, like a bird of paradise, you've probably all seen on BBC or whatever, with this huge plumage in the displays, that bird is balancing a lot of factors. It's trying to be seen, it's trying to get noticed, right, by its female. It's trying to not be noticed by its predator, to be consumed. It's doing a lot of things, and it has to do it in the right place, in the right time, and in the right way. When scientists like myself were trained going through PhD, we would go to the museum, we would measure them with spectrophotometers in a particular place, but that wholly underappreciates the entire complexity of the signal. It underappreciates how it's being used, when it's being used, and how it's being perceived. So what we're interested in doing is sort of moving beyond those types of techniques and using photography, and we can take calibrated photographs that allow us to project something as another organism would see them, like, for example, the female of that bird of paradise. But the challenge with doing something like that is that you can't on the fly change your perspective. You know, you have a snapshot of like a scene, so it's a lot better than the spectrophotometer was. So what we're doing is characterizing that in sort of a video aspect. So a filmmaker could actually go out and take those kind of calibrated videos. Those videos then can show that whether you want to change it in sort of real time for your storytelling perspective, tell it as the eye of the predator, eye of the parent, like the female, or for example, like if they want to avoid a parasite, you could change it on the whim. And so we're sort of trying to connect science communication, but also answer these questions that sensory biologists really haven't been able to ask and certainly haven't been able to answer about how they're using space and time for the signal is chose to be displayed at a certain place and a certain time. So like you could be a not so sexy male, but you can display really well and in the right place, and you can outcompete all of your competitors because of that. And so those are the kinds of things that we can't deal with. But now, you know, the way that we're calibrating these cameras, the way that we're actually splitting light and approaching the question allows us to be able to begin to answer those kinds of questions. And so students are sort of chipping away at various aspects of that currently. I'm hoping to have some present at there's a SciTech conference, like a poster conference. So some of them are going to have their mini projects, which are sort of slowly chipping away at that question to sort of develop that tech at various scales.
Awesome, thank you.
Can I add one thing? For me, so when I was a PhD student, undergraduate, when we were doing any work on molecular biology, looking at how organisms respond, the technology was so backward compared to now, that we used to like amplify one single gene. And that scene, when you got that gene, you were like, wow, I got the gene, amazing. And in my career, I went from amplify 1 or 2 genes to sequencing entire genomes. And not only this, but you can actually go from genome to, you know, all the chain, to the transcriptome, so the gene expression, to the proteins, to the organism. And the power of that is so amazing. And so and that even like is not that expensive, like, okay, it's expensive, but not like that expensive. We just bred some gecko, we did some control breeding, and then we basically just did some deep sequencing, and now we are gonna hopefully be able to link like the gene to the effect that we see in the organism and all in between. And this technology, not even to talk about CRISPR-Cas, but like this technology is so amazing, and it happened in my lifetime, and it's to me it's unbelievable where molecular biology is going. It's just unbelievable, and you can use it for, you know, as a background for all the different fields that people work. It's just amazing.
That's awesome. It looks like we have a question from Aisha.
Oh, hello. Thank you all so much for sharing, you know, your research and everything. It's been super interesting to hear about. I personally don't have a lot of research experience, unfortunately, due to COVID. I'm like a third-year student, and any research plans I had fell through, unfortunately. But it's been really nice to hear about this stuff. But I think, I mean, this might be a little bit of an ignorant question, but I've always just been curious, like, you know, especially when you're starting out in research, like, how do you identify that there's a gap in research in something? Because I feel like everything or most things that are studied are so specific. So like, how do you even come up with that idea of, you know, this is something that needs more research on, and this is something I want to pursue. So just kind of getting started in whatever specific field that you're studying, like how do you come up with the question or the topic or the whatever category of research that you want to focus on?
Am I going to be the first one to jump in again?
Oh,
Someone else go first.
I would say read and talk. So do a lot of reading. So get excited about something you actually care about. Do a lot of reading about that. I would say ask some questions and reassess. And then as my other token that I mentioned before is to not be talked out of something necessarily. So at that reassessment stage, somebody might be so entrenched in the way that the field has been for a very long time that you actually might be onto something very interesting. And so read, talk, think, reassess, and then you might abandon that particular idea and say, okay, well, now I've learned something, and move on to the next. Or you might say, you know what, I think that this is actually interesting, and I'm bringing something new to the table. But you really do have to go through some of those initial stages of reading, thinking, talking, and assessing. But then if you're actually onto something interesting, don't be talked out of it, because sometimes people get so focused on what they're doing and so narrow-minded about their field that they might not appreciate how interesting your ideas actually are.
I'll jump in and totally back up that point about reading. So the first thing is to find out what has been done. And I find a lot of students are good at that. They know to go and read. They've learned how to like write a paper on a topic and find out what's been published on that topic. And I see a pattern where sometimes students stumble across an unanswered research question and don't realize it at first. And it happens like this. They have something they're interested in, and they're going to do, say, a class presentation on it or a class paper, and they go to look for papers on the topic, and then they'll come back to me and say, for some reason, I'm not finding any papers on this topic. And they think they've done the literature search wrong, that there must be papers out there that they haven't found yet. Usually, in fact, there are no papers because nobody studied it yet. And that is your research question. That question you tried to do a paper on and couldn't find anything on, that's your research question. And I find actually a lot of students have had that happen and haven't realized they have identified a gap in knowledge right there. So that's one good place to start, is notice times when you couldn't find papers on something. Another thing is when you are doing your reading, suppose you have found some papers, notice at the end of the discussion, often the authors of a paper will say, well, we have answered this question, but we still don't know these 3 things. And the next step should be this. They are laying out an open research question for you right there at the end of the discussion, and pay attention to those moments too. Sometimes it'll get you pointed toward an area where you hadn't realized that there were still unanswered questions.
Thank you so much. It was very insightful.
It looks like we have a question in the chat. What would you say are the enjoyable and challenging parts about being a researcher in general? David or Dr. Luther has already answered in the chat, but does anybody else want to talk about what they find enjoyable or challenging?
challenging. A lot of things can be challenging. One of the things that is challenging is when you have this brilliant, or at least you think it's a brilliant idea, or like a very interesting idea, but you just cannot figure it out how to do it. Like, you know what you want to do it, and it's great, and you have all the pieces, but then there is one pieces that of the puzzle of the experiment that you actually don't know how it's gonna work or how to do it, and you spend months doing. Like, I'll give you an example. So right now we have this experiment on the geckos and how they respond to temperature. Well, some of my gecko climb, and we need them to not climb to do this experiment, because otherwise every single time we open the terrarium, they just escape. And so that's a relatively simple things, right? Well, it's 2 months we try to get the gecko to not climb. And so we try like oven paper, the one that you put in the oven, the baking sheet, and they climb on it really well. Other people said, try the oil, they climb on it. Other people said, try the whiteboard, and the gecko just climb on the whiteboard. So it's like 2 months that we are trying to get the gecko that climb to not climb to actually be able to do the experiment. So, unexpected things, yes.
Sounds like a gecko jailbreak.
This is for Dot Herling. The gecko can stay upside down on just one finger, one toes actually. So actually they're really funny. Some gecko is on the ceiling on one finger, on one toes, and then all of a sudden they decide to drop. And if you're under it, they drop on your head. And yes, and it's funny because in Asia there is this big gecko, the Tokai gecko, and I know the wife of one of my friends, she's from Thailand, and she's deadly scared of gecko because when she was a kid in Thailand, it happened to her that she was combing her hair, and then one of these gecko was on the ceiling, and all of a sudden he decided to just let it go, and it fell on her head, and this was a little kid, and since then she's like deadly scared about geckos. So no, tilt doesn't work.
Just a quick, I had experience with gecko too that as a kid, I drank my milk, maybe left on the table and found a gecko inside after I finished the milk. That was horrible. But just to answer the question, I think some of the challenges for me is to be able to switch my mind between sort of, if you would imagine sort of fighting in the trenches, troubleshooting minor things, and then also switching your mind to thinking more broadly about the general question. So just being able to do like both at the same time, maybe in the same day, is kind of difficult at times. Sometimes you get so bogged up, looking at computer codes or looking at lab protocol that you get lost among the trees. That's one of the challenges.
Does anybody else have any other questions or comments? All right, if not, what I'm going to do is I'm going to present to you guys a little bit about oh, wait, one more question. Sorry, before I even do that. If a student has an idea for a research project, what are some ways that they can share the ideas with others? This is from Anna Klotzko.
Well, if there's someone in the department that does anything related, they can reach out to them and talk about it. That could be a first stop. Or then, of course, just look and see if things like that have been written by looking in journals and Google Scholar and things like that also. Those are the two low-hanging fruit options I see.
And to piggyback off of that, as a staff member in the department, you can always reach out to us at biology@gmu.edu or ask any of the people you see working around your labs and your classes or your GTAs to see if you can pick their brains to see if they know of somebody who's related. It's basically just creating your web of networking.
And also Oscar, you could, apply to that and sell your ideas, because Oscar is based on you, and then you can sort of bring in your idea and sell it to different professors, maybe if you have time and space for you, because you're bringing your own sort of funding to that undergrad project. Sorry, I think I interrupted somebody.
No, that was perfect. Thank you.
I would just follow up and say, if you have ideas or things you know you're interested in, pursue them. You know, once you graduate, you'll be working for a lot of years. Might as well be something you enjoy.
And isn't that why we're all here? Because everybody has a passion in biology. This is the start of it. This is the seed. Got to let it grow. Any other questions before I go in? I'm going to talk a little bit about the biology research semester. All right, well, I'm going to share my screen. If you do have questions, put it in the chat, and then we'll have some time a little bit afterwards to talk about it. Okay, so I'm going to share my screen. Okay. So if you are interested, you're an undergraduate and you're trying to get more experience in the research semester, it's probably why you're at this presentation, amongst other things. You've probably heard about the biology research semester. So what I want to share with you guys is what the research semester is. Semester-long program happens in the fall every year. Well, mostly every year. COVID had other answers for last semester. But it's a semester-long program that allows students to get involved and immersed in an ongoing scientific research at Mason. You register for full-time work side by side with working researchers. A lot of the panelists today are actually part of the research semester or may become part of the research semester in the future. Who knows? You work in the lab, in the field, online, or a combination of the above, like we were talking about earlier. It's a really, really fun program where you take time out of a standard semester where you go class to class and you actually actively work on something that you formulate from the ground up. So you'll be immersed in a full-time research project while earning 12 to 15 credits. You'll have the chance to network with distinguished research faculty, hint, hint, nudge, nudge, everybody in this room, and research labs in the area of your interest. You'll have the opportunity to present your research in public, so you get some of that networking opportunities and presentation skills. And that's not only at Mason, but at regional or national scientific meetings as well, especially with our Oscar program, who works, and students and scholars programs who work with us to fund your research. So you are funded when you become part of the research semester. You get a little lump sum and you get to buy supplies and do all the things. You are being put into the field as a full-time research semester for the fall semester. Who would you work with? That's part of the networking that I was talking about earlier. You pick in the application process, you pick your top 3 choices from a list that we provide at the department. This is available on our website. And you go through an interview process with those researchers. It's not only them picking you, it's you picking them. We have a variety of experts and unique topics across the College of Science. You can see a lot of the people we collaborate in this graph here. And this is just a very limited list. We have plenty of other opportunities available that aren't Mason involved. We have, different Quest and all these different companies who collaborate with us as well. So it's not just limited to this list, and it's pretty awesome who we work with. What does your schedule look like? So this is a full-time program. So it's 12 to 15 elective credits towards your degree. So you would not be allowed to take more than 1 class in addition to the research semester. So we recommend you keep your schedule clear because we want you fully immersed in this process. You'll take 6 to 9 credits of research with a faculty member. Then you'll take 2 classes on top of that, research seminar, which is 2 credits. and Introduction to Research Design and Analysis, which is 4 credits. And the reason we want to keep your plate clear is because you have to dedicate at least 27 hours per week for these research projects. But in the long run, this will actually work out really great, because at the end of the semester, we've got all these different things going on. So one of the big things is, while we don't provide scholarships, this is a normal semester, so you pay as usual. It's normal class registration, except for the research, which you would have to do a different kind of process, which you'll get covered when you get in. But we do have a biology research celebration. So at the end of the semester, we have this big thing over at the Center for the Arts typically, but we're probably going to be looking around for next year. And you present your work to a panel of judges, and at the end of this, the judge will pick on specific criteria who the winner of the competition is. And this basically gives you the Biology Research Semester Award, which will be $500 and 1 to 2 honorable mentions for around $200. And this is a award you can actually receive at graduation. So we have at least 3 winners every semester for Biology Research Semester Award. And then we have the Marion Lobstein Scholarship Award. So that one is even more special. So that's given to a rising senior with the best project at the research semester celebration. And that gets a lot more money. I can't really elaborate on that. I didn't write the number in the slides here, but you get to present at the Virginia Arts and Science Conference, and you get a lot more perks with that. You get a scholarship towards your senior year at George Mason University. And so that's just a quick overview of what the research semester is. It's definitely a fun and fulfilling experience, especially if you want to deep dive into what research is. You can go about it on your own. You can go through Oscar. You can go through all these different things, but why not try and join us for our biology research semester? I've heard so many good things about it. Students love the benefits. I mean, it looks great on your resume. It gives you all the experience you need to be prepared if you want to go into grad school environment of research. So if you want to apply, if you're interested in this at all, go ahead and go to this website here, tinyurl.com/research semester, and you have until March 31st to apply. So I highly recommend it. If you don't want to do it, that's fine, but at least you can check out the list of research projects and faculty mentors, and you'll get actually more in depth on our presenters today. And I hope to see if you guys apply soon. Thank you. So on that note, let me see, I see 2 questions. For the biology research semester question, we don't have it in the spring. We were planning on holding it in the spring this semester, but because we were still semi-shut down, we could not run it in the spring. We have to have some in person. Most of our research is in person. So we want to make sure that we're going to follow our same schedule and keep it in the fall. So fall 2021 will be the next time it's offered. And as for group research, we want to make sure that this project is solely you, but researchers tend to pick 1 to 3 people to join their labs. So you will be collaborating with other students who might be in the research semester, but at the same time, it's not a group research effort. You will be providing your own project and your own take on the research that you are doing. So is there any other questions on the research semester or for our panelists? I hope that came off clear. I hope everybody was, if you have any more questions for me, obviously, let me know.
I saw one question about whether there could be group projects. Sometimes you want to work sort of collaboratively in a team, and a lot of times you can sort of when you're working in a team, you can fit a variety of very independent projects together in a way that essentially you all are working as a collective whole, but you each have your own independent project and that are sort of fitting together as puzzle pieces. And so all of us can help you sort of find your piece in that puzzle. So if don't be afraid if you kind of want to have more a larger support system of working in a group. That happens even if it's not immediately apparent when you see the posters. That can you can arrange that and people will work together at various aspects. So just I want to point that out.
Well, I want to thank all of our panelists. We have another 10 minutes, so if anybody wants to, or I'm sorry, I think we were supposed to end at 11:15, but I'll keep the chat and everything open for another 10 minutes. If you have any questions or we want to say anything. In the meantime, though, I'm going to share with you guys our panelists today, all their email addresses. If you want to talk more about research semester, you email this. I think that I actually misspelled the email for research semester. I'll correct that in the chat. And if you have any questions about biology in general, just email us at biology@gmu.edu. Any of our panelists? Oh, thank you to all of our panelists. If anybody wants to stick around, that's fine. But otherwise, please send us an email if you're interested. And if you want to see more events like this, we should be having more soon. This is definitely what we want to be doing more of in the future. So if you have any ideas or anything you want to contribute as a student, please let me know. And you can email biology@gmu.edu any ideas you might have. I'm totally open to everyone's opinion on this.
Thank you, Genevieve. This is great.
Thank you, Genevieve. Thank you, everyone. Good to see you.